Thank you for joining our webinar today. Today's webinar is an introduction to the In-Cell Western Assay. Our speaker is John Lysate, Field Application Scientist at Lycor. Please introduce yourself, John.
Thanks, Ashley. My name is John Lysate, and I'm a Field Application Scientist with Lycor Biosciences. I cover the New York City metro area as well as Connecticut and New Jersey. Prior to joining Lycor, I was a postdoctoral fellow at the New York University. I obtained a doctorate in pharmacology at the University of Washington in Seattle, Washington. During my doctoral work at the University of Washington, I did extensive work with the In-Cell Western Assay and using this assay to study signal transduction activation following additions of ligands or inhibitors to G-protein coupled receptors.
Thank you, John. Our panelist today is Margaret Detlinger. She will be answering any questions that John doesn't get a chance to answer during his talk. Margaret, please introduce yourself.
Hello, everyone. I'm Margaret Detlinger, Digital Media Product Manager. Please feel free to tweet about this webinar using hashtag RethinkWesterns. We look forward to your comments.
Thank you, Margaret. Now let's turn it over to John. John?
Thanks, Ashley. So today what I want to talk about is the In-Cell Western Assay. I want to cover exactly what is the assay, what are the benefits to using this assay over a traditional Western blot, and what are the different type of experiments or applications you can use this assay in. In addition to the In-Cell Western Assay, I also want to talk about a variation of the assay called an On-Cell Western Assay. And then finally, I just wanted to summarize everything I talk about in a case study in which a group looks at Alzheimer's disease and uses the In-Cell Western to screen compounds for treatment in Alzheimer's. So let's first start with talking about what exactly an In-Cell Western Assay is. An In-Cell Western Assay allows you to approach your science and approach your experiments in a high-throughput manner. By using an Odyssey system from Lycor, it allows you to simultaneously detect and quantify two separate proteins at once within a cell. The Odyssey systems, specifically an Odyssey Classic, an Odyssey CLX, and an Odyssey SA, work with the In-Cell Western Assay. All of these systems have two solid-state lasers that excite fluorophores at 785 and 685 nanometers. Because we have two solid-state lasers at different wavelengths, this allows us to simultaneously detect two proteins by using two separate secondary antibodies. Each of these antibodies will have a unique infrared fluorophore specific to each laser. Now let's walk through the protocol. An In-Cell Western Assay starts by simply plating your cells in a microtiter plate. These plates can range from 6-well plates all the way up to 1500-well plates. The only requirement in these plates is that they're clear-bottomed. Now these cells can be suspension cells or they can be adherent cells. Now once you're done plating the cells and you've allowed the cells to grow to confluency, you then do your treatment. Now depending on what you're interested in or what you're studying, these treatments can vary. Perhaps you're looking at a signal transduction pathway and you want to add a ligand of interest. Perhaps alongside your ligand you want to add an inhibitor and see how an inhibitor affects the pathway. If you're doing siRNA screening, you can add siRNA to each of these wells. Depending on the size of the well, you can change the conditions from well to well. Once you're done with your incubation, you remove the media, you fix, and then permealize the cells. Once you've permealized the cells, you go ahead and block each well, add your primary antibody, wash, secondary antibody, and wash. Again, as I just described, what makes these secondary antibodies unique is that they will be coupled to an IR dye. These IR dyes will allow for detection on an Odyssey system. So once you're done with that last antibody wash step, you can then just take the plate itself and detect your signal on either an Odyssey Classic, an Odyssey CLX, or an Odyssey SA. So at this point, why don't we stop and take a poll? Ashley?
Sure, John. Let me pull the question up. The question is, what type of plates work best for in-cell westerns? You can choose from black-walled with clear bottoms, clear-walled with clear bottoms, or white-walled with clear bottoms. Go ahead and click the radio button next to your answer choice, and we'll give everyone just a couple of moments to choose an answer.
Great. Well, it looks like we have a lot of good answers. Really, with the In-Cell Western, there's no wrong answer here. All three can be correct. What we were trying to get at is some plates might be optimal or better for your experiments and better for your data than other plates. Typically, I would recommend using a black-walled, clear-bottom plate. The reason that I would say to use this plate is black walls in a plate will have less background than a clear-walled or white-walled plate. So the correct answer I would recommend of these three options would be black-walled with clear bottom. Now, let's compare a typical workflow with a traditional Western blot on the left and the In-Cell Western assay on the right. Now, currently in the lab, if you did a signal transduction activation, you do your cell treatments, remove media, lyse the cells, transfer your lysates perhaps to Eppendorf tubes, spin out insoluble material, perhaps fractionate further. Once you have a sample, you have to determine protein concentration, load SDS page gels, run the gels, transfer the gels to membranes, and then you begin blocking in antibody incubations. Each of these steps can be quite time consuming, and so as a consequence, the entire workflow for a traditional Western blot can be quite time consuming. With the In-Cell Western assay, once you're done with that treatment, you remove the media, and then again, you just fix and permealize. Once you've permealized the cells, you go immediately into the blocking and antibody incubation steps. So simply by moving to the In-Cell Western assay, we've cut out a lot of these steps involved with the traditional Western blot. Because we've cut out these time-consuming steps, we save time with the In-Cell Western assay. Now, one important thing to distinguish between these two assays is your antibodies you might be using right now for your Western blotting might be recognizing a denatured state protein. One important thing to keep in mind as you move to the In-Cell Western assay is you want to make sure your primary antibodies will recognize native state proteins. So we're going to save time in just the workflow and cutting out some of these steps. We're also going to save time in just the amount of data we get. So if we did an In-Cell Western plate and we used a 384 well plate, that's 384 different data points we can get at once. If we tried to get that same amount of data using traditional Western blots, we would have to run roughly 43 10-well gels. So we're not only going to save time in cutting out steps in the protocol, but we're going to save time in how much work we have to do to get this data. Now, not only do we save time, but we get better data. So what I'm showing here is two graphs. The graph on top is a graph from an In-Cell Western experiment, and the graph on the bottom is one from a traditional Western blot. Now, the group that generated these graphs did the same exact experiment. They did it with an In-Cell Western on top and a traditional Western blot on the bottom. Now, the specifics of the experiment aren't really important here. When you look at this data, the key things to understand is that this group was screening 20 different mutants. The hypothesis was that these mutants would increase the phosphorylation level of a target protein. And it would increase this level of phosphorylation to different degrees, depending on which mutant you looked at. So whether they did a traditional Western blot or the In-Cell Western assay, they were quantifying the phosphorylation level of their target protein. Now, there's two key things that when you look at this data. The first thing to keep in mind when you look at the data is the trends are very similar. When you see high levels of phosphorylation with a Western blot, you see high levels of phosphorylation with an In-Cell Western. Conversely, if you see low levels of phosphorylation or no phosphorylation with a traditional Western blot, you see low levels of phosphorylation or no phosphorylation with the In-Cell Western. So we're not changing the data we're getting. The key difference between these two graphs is the error bars. If you just look at the two graphs, the error bars are much tighter with the In-Cell Western assay. In fact, asterisks in these graphs are depicting significant results, meaning significantly higher than controls. When they were screening these mutants and screening for mutants that were significantly higher in levels of phosphorylation above controls, with a Western blot they could only find 4 mutants that were significantly higher than controls. Now again, same experiment, but with an In-Cell Western assay, they were able to find 15 mutants that had significantly higher levels of phosphorylation. So not only are we saving time, not only are we getting a lot of data quickly, but it's better data. And the reason why you have tighter error bars is because we've increased the reproducibility from experiment to experiment, from well to well. We have achieved this reproducibility because we removed a lot of the steps involved with a traditional Western blot. I just talked about the workflows. With a Western blotting, you're lysing cells, spinning out insoluble material, running gels, transferring those gels. There's a lot of steps there. One, with liquid handling, where you might lose sample, change sample to sample. You might be losing protein along the way. All of these steps increase your variability from experiment to experiment. Because the In-Cell Western assay has so few steps, since you only fix and permeabilize the cells and then begin your antibody incubations, we've removed all of those chances to induce variability in the experiment. So we have better reproducibility, tighter error bars, better results. So at this point, I want to just go through and give an in-depth example, or walk through an in-depth walkthrough of how you can use the In-Cell Western assay for the experiments you're doing right now in the lab. So to begin with, I want to talk about protein phosphorylation. So in this example, we have our cell of interest, and let's say we've plated them in a microtiter plate, like a 96-well plate. And these cells of interest are expressing a receptor that we're studying. So once we've seeded our cells and they've grown to the desired confluency, in this example, we are going to go ahead and add a ligand. These ligands will bind to the receptor and activate those receptors. Once activated, downstream pathways will also become activated, and we will have phosphorylation of our target protein in this example. Now, depending whether you're doing a dose-response curve or a kinetic assay, you can change the concentration of your ligand from well to well, or you can change the incubation time from well to well. Once you're done with the incubation, you remove the media again, fix the cells, permeabilize, and then move on to the subsequent steps. Now, in this example, when we add our primary antibody, the primary antibody will be specific for our phospho target. We wash, add secondary antibodies, the secondary antibodies being specific for our primary antibody. Now, again, what makes these secondary antibodies unique is that they're conjugated to an IR dye that will allow for detection on an Odyssey imager. So at this point in the example, once we've incubated the cells with our secondary, we can go ahead and go to our Odyssey and scan the plate. Now in this example, I'm only using one primary antibody, detecting my target of interest, in this case phosphorylation. But there's a key thing to keep in mind is, well, what do we need to look at in the second laser channel? And what we need to look at is normalization. What we need to do is we need to normalize the level of phosphorylation from well to well to a loading control, if you will. Now, it's important because if we have differences in cell numbers from well to well, we need to account for that. There are a lot of different options out there to choose what should I normalize to. The first two options are typically let's normalize to a total protein or let's normalize to a housekeeping protein. So in my example, I'm looking at a phosphoprotein. So if I had an antibody against the total protein, then I could normalize in my second laser channel to the total signal. If I don't have a total protein antibody, perhaps I want to choose a primary antibody against GAPDH or actin or another housekeeping protein. Now, these first two options require the use of primary antibodies and secondary antibodies, typically which can be quite costly. And additionally, if you need to optimize these conditions, it can take a lot of time away from other experiments. So the third option I want to talk about is normalizing to cell number. In this example, or for this to work, you simply just use non-specific cell stains that will just stain the entire cell. So in one laser channel, we will have cell number, and then in the second laser channel, we'll have our target of interest, in this case, our phosphoprotein. So if there are differences from well to well, our non-specific cell stain will show us those differences, and we can normalize to those differences. Now, before I talk about the different staining options out there, at this point, why don't we go ahead and take another poll? Ashley?
Our second question is, which channel is best for normalization? You can choose the 700 nanometer channel or the 800 nanometer channel. Go ahead and click the button next to your answer choice, and we'll give everyone just a couple of moments to finish.
Looks like we're getting a lot of good answers. Now, of the two options, what I'm looking for here is I would choose to normalize in the 700 channel or the 700 nanometer channel. The reason for this is that if you compared the two laser channels, since the 800 nanometer channel is deeper in the infrared wavelengths, you have less autofluorescent properties of your tissue and the plate. Because you've removed these autofluorescent properties, you have much greater sensitivity in the 800 nanometer channel. So if I'm looking at a phosphoprotein, in my example here, I will be able to see lower levels of phosphorylation in the 800 channel. So if in the 700 I use a non-specific cell stain, in the 800 channel I can look at my target of interest. So let's talk about the different options out there for these non-specific cell stains. Now, you might have seen some of these stains out there. I'm listing just a couple here. Very commonly, you might see DRAC5. DRAC5 is a non-specific DNA stain, but it works best when you have lower cell densities. Another option, Sapphire 700. Sapphire 700 stain is a non-specific nuclear and cytoplasmic stain, but it only works at higher cell densities. Now, if you look at these two stains and plotted them against cell number, shown here, you'll see the differences in linearity for these stains. So there's three traces here. The bottom trace is Sapphire 700 alone. The middle trace is DRAC5 alone. And then the top trace is the combination of the two. So what you can see is Sapphire 700 has very poor linearity over the entire range, and again works best at the higher cell densities. DRAC5 only works well at lower cell densities and plateaus off as you get to higher cell densities. So the workaround to using either of these stains separately to have it work at any cell density is to combine the two together. So in the top trace, when you combine the two, you have linearity for the two stains together over the entire cell range. Now, a lot of groups don't like to buy lots of cell stains because the goal of using these non-specific cell stains is you don't have to get a lot of reagents and spend a lot to get these results. So rather than using both these stains and trying to optimize each condition separately and then together, at Lycor we offer CellTag 700 stain. This is a one stain solution that works at both the low and high cell densities. and it's a nonspecific nuclear and cytoplasmic stain. So if you use cell tag 700 stain, it gives you linearity and a nice tight fit in this linearity. Again, the benefits here is it's a one stain solution, requires very little optimization, and it's highly affordable for lab purposes. So let's go back to my example. We're going to look at phosphorylation of a protein target. So what does the data exactly look like once you've scanned it on an Odyssey imager? So in this example here, I'm showing 3 images. It's a 96-well plate. In green, in the middle image, is my 800 channel signal. Here I'm looking at my total protein. In red is my phospho signal, or my phosphoprotein of interest. So in this example, I'm looking at the EGF receptor. And I'm looking at phosphorylation of the receptor after addition of EGF. So from left to right in these wells, done in duplicate, is a steady concentration of EGF. Then starting in column number 3, as you move left to right, there's a decreasing concentration of inhibitor. So what you can see is when you add EGF alone, we get high levels of phosphorylation as seen in the 700 channel. When we add a high level of inhibitor, we block that phosphorylation, and then as we titrate out the inhibitor, we regain levels of phosphorylation. Now with Odyssey software and Image Studio software, you can either look at these two signals separately, as shown on the bottom, or you can overlay the two channels and easily tell what is phosphorylated and what's not. In addition to being able to view these images so easily, Odyssey software and Image Studio software allow you to easily apply grids on top of these images and adjust the grids for your own specific plates. Once you've applied these In-Cell Western grids, you can export the data to a graphing program of your choice. So in this case, if I'm looking at an inhibitor of my phosphorylation event, I can easily plot the data and then determine what is the IC50 for this inhibitor. So let's talk about another example. Let's transition from protein phosphorylation, and then let's talk about instead protein trafficking. So in this example, I'm going to again talk about the In-Cell Western Assay, but I'm going to compare it to the On-Cell Western Assay. So in this example, what I'm most interested in is receptor localization. So where is my receptor inside a cell? Is it at the plasma membrane, or is it internalized in an internal structure? Now in this example, I'm going to have 2 side-by-side wells. One well will be shown on top for the In-Cell Western Assay, and then the well next to it will be shown on the bottom for the On-Cell Western Assay. Now if I'm going to do any cell treatments or anything like that, both wells will be treated identically. So once I'm done with my treatments, if I'm doing any treatments, I again remove my media and fix the cells. Now with the In-Cell Western Assay, I would say at this point you would permealize the cells. Now the On-Cell Western Assay is different in this sense. The In-Cell Western Assay we permealize the cells. On-Cell Western Assay we do not permealize the cells. And this is the key difference between these two protocols. So when we move forward with the assay and add our primary antibody, since the In-Cell Western Assay cells are permealized, our primary antibody will be able to find epitopes that are both external and internalized. The On-Cell Western Assay, since these cells are not permealized, our primary antibody will only be able to bind to extracellular antigens. So with my In-Cell Western Assay, my primary antibody will bind to receptors localized at the plasma membrane, as well as any internalized receptors. And then with the On-Cell Western Assay, my primary antibody will only bind to receptors localized at the plasma membrane. So I can add my secondary antibody, again being conjugated with an IR dye to allow for detection on an Odyssey system. I can then scan them, quantify my results, and I can compare my results from the In-Cell Western Assay to the On-Cell Western Assay. So by doing these side-by-side assays, we can address some different questions. We can look at protein trafficking. Where is your protein at inside a cell? Is it at the plasma membrane or is it internalized? Doing the In-Cell Western Assay, we can see how much of our protein is expressed throughout the cell, and then the On-Cell Western Assay, we can see how much of that signal is perhaps at the plasma membrane. Alternatively, using the On-Cell Western Assay, we can look at receptor internalization. So perhaps we start at a basal state and we can assess how much of our receptor of interest is at the plasma membrane. And then we can add ligand and see what happens to our signal after we add ligand. So if our receptor is internalized and we do an On-Cell Western Assay, we would see our signal go away. Additionally, we can do receptor recycling. So after our receptor is internalized, when does it come back to the plasma membrane? Now, there are a lot of different In-Cell Western applications, and I don't have the time today to go through each one of these. They're listed here, just some of the applications you can use this assay for. Gene expression, viral titer, load, apoptosis, RNA screening, cell proliferation. There's a lot of different applications out there for this assay. And again, rather than spending a lot of time to go through each of these and how you can use it in the lab, I encourage you to go to lycor.com/ICW. At our website, we have a ton of information about this assay. We have overviews, we give protocols, example publications. Additionally, we have links so you can contact us, and we can get you in contact with your local field application scientist, and they can help you develop this assay as well. So let's talk about how a group has used the In-Cell Western Assay in their own research, and specifically with Alzheimer's disease. Now, just to give a little bit of background for Alzheimer's, since we have a diverse crowd here, Alzheimer's has a couple different hallmarks. The main biology hallmarks, when you look at an Alzheimer's patient, is they have A-beta 42 plaque formation, as well as hyperphosphorylation of tau. Current treatments for this disease are mainly targeted at improving the memory function. And because we have this plaque formation and hyperphosphorylation of tau, there's a lot of neural toxicity. So giving patients NMDA antagonists to try to decrease this toxicity. Now, both these treatments don't get after the hallmarks of the disease. So they're not a cure. They're mainly trying to improve quality of life. So in efforts to find a cure, groups have looked at two main targets, again with the A-beta 42 plaque formation and tau hyperphosphorylation. With the A-beta 42 plaque formation, groups have focused on let's block this plaque formation. What was found that even if you block plaque formation, tau dysregulation alone might be sufficient and can be sufficient for the disease. So in efforts to also look at tau hyperphosphorylation, groups have looked at tau kinase inhibitors, microtubule stabilizers, and aggregation inhibitors. But with each of these drugs, there have been issues. So with a kinase inhibitor, you might be able to block one tau kinase, but there are a lot of tau kinases in the cell, and so blocking one is not sufficient to stop tau dysregulation. So the question was asked, rather than trying to target and block this phosphorylation itself, what if we use the biology of this cell and increase chaperone expression? And the idea being, perhaps if we increase chaperone expression, these chaperones will decrease tau levels and block this hyperphosphorylation. Now, the proof behind this question, or the proof behind this hypothesis, was that years ago, a compound was discovered called geldinomycin. Now, when looking at geldinomycin, it was found that the main function of this compound was that it would inhibit HSP90. And what was found is that once it inhibited HSP90, cells would increase overall chaperone expression inside the cell. Then what was found is after you had an increase in chaperone expression, tau levels would then decrease. So it looked like a great promising compound that you could maybe perhaps use for treatment of Alzheimer's. But the issue with geldinomycin is that it can't cross the blood-brain barrier. So in a patient, it can't get to where it needs to be inside the brain. So Dickey et al. In 2005 asked a question. Let's look at HSP90 inhibitors, and let's screen compounds that may inhibit it and can inhibit HSP90. So what they did is they did the In-Cell Western Assay. They treated cells for 48 hours with 1 micromolar of these different compounds, and then assayed for HSP90, chaperone expression, and tau levels. So here's some of the example data that they found. What I'm showing are the In-Cell Western data, and alongside I'm showing the Western blots that they did as a proof of concept. So let's just start at top, looking at total tau protein. Again, we have 10 inhibitors, A through J, and then a vehicle-treated control. So when they incubated these cells in triplicate with these different compounds, what they found is that these different compounds had varying abilities to decrease tau levels compared to control. The middle image pane is GAPDH. In this example, they're using GAPDH as their normalizing signal. The bottom image pane is looking at HSP70. So again, the idea here being that these different inhibitors, A through J, will inhibit HSP90. Once HSP90 is inhibited, you'll have a correlating increase in overall chaperone expression, such as HSP70. So when we look at HSP70 signal, we can see vehicle-treated controls have low levels of HSP70 expression, and then these different inhibitors have varying effects to increase the levels of HSP70. Now, again, using Odyssey software and Image Studio software, we can quantify these results and plot them. And so what I'm showing here is tau and GAPDH. Tau in black, GAPDH in gray. And again, what we can see is that these different compounds, A through J, had different effects on tau levels. Some inhibitors, like inhibitor F, greatly decreased tau levels. Some inhibitors, B through E, had a more moderate effect, and then other inhibitors didn't really change the levels of tau. If we look at our HSP70, we can see again that these different inhibitors had varying effects on HSP70 expression levels. So the main conclusions from this data was that they found that HSP90 inhibitors can decrease tau levels with varying effects. There's also a correlating increase in HSP70 expression. They also found, and I didn't show the data here, but they also found that there was increases in HSP27 and HSP40 expression with these inhibitors. Now the exciting thing here is that these inhibitors had a chance that they may be able to cross the blood-brain barrier. So there's a huge therapeutic potential with these drugs. Now, to bring it back to the In-Cell Western, the key thing found here was that the In-Cell Western allowed for this group to quickly screen these compounds. Again, depending on the size of your wells, whether it be 96 or 384, you can quickly screen a lot of different conditions and get high-quality data. So to summarize, the In-Cell Western Assay can serve as an alternative to some of your traditional Western blot experiments. It'll save you time in the protocol, save you time in just getting sheer data collection. Since you're using microtiter plates, you don't have to use nearly as many reagents as with a Western blot. And overall, you get better reproducibility, less variability, and better data. And I would encourage you, for more information, to visit lycor.com/ICW and learn more about this assay, and you can download protocols and see how you can use it in the lab. Ashley, do we have time for some questions?
Sure, John. I've saved just a couple of questions for the end of the webinar. The first question I have is, what if my primary antibody has nonspecific bands?
That's a really good question. If your primary antibody recognizes a lot of nonspecific bands on a Western blot, so let's say it recognizes your band of interest, but also 10 other bands. If you transition that experiment into an In-Cell Western, that means the signal you read on an Odyssey, a fraction of it will be your target of interest, but a majority of that signal you read in the well will be nonspecific. So it will decrease your sensitivity and decrease your ability to detect changes in your target of interest, whether it be phosphorylation or protein abundance. What I would recommend is, if you do have a primary antibody that's nonspecific, that you optimize the primary antibody conditions to decrease the level of nonspecificity. This can be achieved with blocking conditions, detergent conditions, and I would encourage you to work with your local field application scientist, and they can help you also to increase the specificity of your primary antibody. Ashley, do we have another question?
The second question is, my primary antibody states it is compatible with immunohistochemistry. Will it also then work with the In-Cell Western?
Yes, you're totally right. If your primary antibody states on the data sheet that it's compatible with immunohistochemistry, then it should also be compatible with the In-Cell Western assay. When a primary antibody states that it works with immunohistochemistry, what it means is that it can recognize a native state protein. Again, the key differentiating fact here between a Western blot and the In-Cell Western assay is your primary antibody needs to recognize a native state protein. So if the data sheet does state that, you should be good to go with that primary antibody. If it doesn't state that it works with immunohistochemistry, you might want to contact the antibody vendor and see if they have more information regarding the primary antibody's ability to recognize native state versus denatured state proteins. Or if they don't know, or if it doesn't state it, perhaps you want to switch your primary antibody to a different primary antibody that will work with native state epitopes. Ashley, do we have time for one last question?
Sure, John. Our last question is, can I just use my plate reader to read these plates?
That's a great question. I get that question all the time. Plate readers typically are in all the labs. The issue with trying to do this assay with a plate reader is plate readers only take a couple samples per well. An Odyssey system will scan the entire surface of the well. So if your well is not 100% confluent and you put that plate into a plate reader, if the plate reader tries to excite these fluorophores and it hits a spot where there are no cells, perhaps you're not at 100% confluency, maybe you're at 70% confluency, and in a given well, a plate reader might miss signal. Since the Odyssey system and Odyssey imagers scan the entire surface of the well, even if you don't have 100% confluency, we will gather the entire signal from that well. Because we're getting the entire signal, then we will get more information and can have better sensitivity. So I highly recommend that these assays need to be done on an Odyssey imager and not a plate reader. Great. Well, thanks for all the great questions and taking the time to attend the webinar. Ashley?
Thank you, John. I think this is a good time to tell our audience that Lycor also offers a custom service program where we can optimize the entire In-Cell Western protocol for you. All you need to do is send us your cells and primary antibody, and we can determine all of the appropriate conditions and protocol steps for you. Just visit www.lycor.com/custom services for more information. I'd also like to thank our audience for taking the time to participate in our webinar today. We will be sending additional information and a link to access a recording of this session very soon, so watch your email or lycor.com/webinars for details on how to watch the replay.